All right. Well, good morning, good afternoon, good evening, everyone, and welcome to the world's most exciting classroom. My name is Joe Grabowski, and I'll be your host for today. Today, we have another action-packed world's most exciting classroom for you. To give you a quick rundown, we are going to catch up with some of the Darwin 200 team on the Oosterschelde in the Pacific Ocean, where they're heading towards Rapa Nui, or Easter Island, as it might be a little more familiar to some of our viewers today. We have another really cool video from the Ocean Conservation Trust at the National Marine Aquarium in Plymouth. We have an amazing experiment, one you can try at home or in your classroom. We've got our Curiosity of the Week, and we're going to meet one of our new researchers. So a little later in the program, we'll meet Jut. Jut will be getting on the ship in a few days in Rapa Nui, and he has a really awesome bit of research that will stretch from Rapa Nui to Pitcairn to Mangareva, which is our next destination after Rapa Nui. So we'll talk to Jut, we'll get to know him a little better, and we'll meet him a few times, hopefully while he's doing some of his research live on some of those islands that we'll be visiting. Okay, so let's get right into it. I have my screen sharing here. I am going to bring in a map. And in fact, I'm also going to bring in our Darwin 200 team standing by. We've got Josh with us this morning. We've got Grant with us. Hey, guys.
Hello.
Good morning.
How are we doing today?
Good morning. It's good. We're surprised how dark it is. It's 7 o'clock our time, but it's still pitch black outside.
Yeah, you guys have definitely pushed a lot further to the west since we connected last. And I know that Easter Island's time zone is probably strange because they want to keep it matched up with Chile on the mainland. So
for sure.
Yeah, very cool. All right. Well, guys, I've been looking at the map here. So let's zoom in here. So here you guys left Galapagos. I think we connected. You guys were somewhere just around here. We saw this first picture of the ship here taken by drone. But then as we kind of follow your path, it looks like you've had some pretty good wind the last few days, or, you know, the last week. There's several pictures of the ship. Looks like you've got the sails up and a pretty good lean going on.
Yeah, there has been some really nice sailing wind. There was a few days where we had all the sails up and we hardly touched them. And yeah, the ship was on about a 10 to 20 degree heel most of the time, which if you're a sailor is great. If you're not a sailor, it's a bit of a pain to live your life at 20 degrees. But then a few days ago, 2 or 3 days ago, the wind died down completely. So all the sails came down and we've just been on the engine since. But we're about to head into what looks like quite a big storm. Or I say a big storm, some quite heavy winds. And the captain seems to be quite excited about it because it's good sailing wind. But that's why we're going off course a little bit, because we're going to try and come in there from the other side and catch the wind going in the right direction rather than heading straight into it.
Yeah, we can see that here. Here's our destination. We can see you guys are a little off course, but I'm sure Jan Willem, our fearless captain, he's got a plan. He's going to get you guys in there on wind. Sounds like it's going to get a little interesting. So hopefully you can capture some video if you guys get some really good wind. And then another thing I noticed is when I was on board Prude Ecuador, we had fishing lines off the back of the boat. We didn't catch anything, but it looks like a different story for you guys. If I click a few pictures here, it looks like you've had some success catching a few fish.
Yeah, at last. We've had a really long dry spell with the ship, with fishing. And it's almost become a running joke that we are the worst fishermen in the ocean. And then one day we caught that big tuna that you just saw before.
Yep.
And we weren't even finished cooking that before we caught the big mahi-mahi the next day. So we had some really nice tuna meals for a couple of days. And I think the mahi-mahi's in the freezer ready to get cooked up at any moment.
All right, getting some fresh fish out there. That's great. Grant, maybe you can talk a little bit to this picture. Looks like there was a little science going on.
Yeah, so we've had a really great bunch of guests this time. Everybody's super curious about all the science that's going on board, and we had some interest in pulling up some water samples to see if we could filter out the water and see if we could find any marine invertebrates that were in the water. During the day, we weren't having so much luck with it. I mean, it's the open ocean, and we're kind of just skimming with a really small net, but I suggested that the guests try again at night, because I knew that a lot of marine organisms are nocturnal and they come up to the surface at night. And that's when you get a lot of the bioluminescence and everything. And so they tried last night, I believe, or the night before last. And in the morning we were looking, they pulled up little copepods, which are little crustaceans with long antennae. And they saw some tunicates, which are they're related to chordates, like us, but they have kind of a jelly skeleton. So they've even gotten some cool stuff, even though it looks like it's just empty blue.
Yeah, we hear a lot about migration of sharks and turtles and whales and birds, but every night there's the world's largest migration, where we have all these organisms that come up and then back down during the day.
Yeah, absolutely.
All right. Well, it looks like you guys have been having a great time. How's the bird survey going, Grant? Any new species?
It's going great. Yeah, we've had a couple of new species. I think the showstopper for us has been tropic birds. Mm-hmm. They kind of look like gulls or terns, but they're these white birds, and all of them, there are only 3 species in the world, and all of them have these long streamer tails that they extend more than like double the rest of the length of the bird behind them. So it's kind of weird because we don't see a lot of extravagant feathers on seabirds, but we've seen all 3 species of tropic birds on this voyage. And that was something that was totally unexpected, but it's just we've covered such a great distance, and now we've started to edge into Polynesia, and so we've gotten 3 out of 3 of the world's tropic birds.
Oh, that's great. Well, there we go. We've got good signs going on. We've got some good wind. We've got some fish. I don't know if the sailing gets much better.
Yeah, well, hopefully once we head into that wind and we get the sails back up, the sailing will get much better again.
Yeah, very, very cool. Well, what's the best estimate? Has Jan Willem given you guys an estimate when he thinks you'll roll up on the island?
The 3rd is the last thing that I've heard.
I think midday, right? About midday on the 3rd. Yeah. So we're a little bit ahead of schedule.
All right, very cool. Well, we're excited about it. We're looking forward to our connection next week. With any luck, we'll have someone running around on the island with a satellite unit, and we'll be able to do something pretty cool from Easter Island. So we can't wait, guys. Thank you so much. I know it's early there for you. Breakfast is probably in about 20 minutes. But thank you so much for giving us that update and letting us visit you out on the Pacific.
No, thanks a lot, Joe.
Absolutely.
Good to see you.
Yeah, take care, everybody.
Awesome, guys. We'll talk to you soon.
Bye.
See ya. All right, so there we go. Let's bring up the map one more time. We are making pretty good progress. You can see where we were in the Galapagos. It was a pretty awesome 3 weeks in the Galapagos, but you can see some good wind. A couple pictures here. There's a picture from the drone. Here's a picture of the sails filled with wind, and I think there was one more down here. No, not that one. There it is. So another nice shot there. The ship on with a nice lean and moving along nicely with some of that wind action. And then there we go, a little off course now, but that's to catch the wind in this system that's moving through and possibly arriving by the 3rd. So we look forward to that. Okay, let's continue on. So like I mentioned, we have another great video from the Ocean Conservation Trust from the National Marine Aquarium in Plymouth. And so we're going to play that video and have a little Kahoot together after the video. So keep your ears out and be ready for a little multiple choice, true and false action. And then by that time, hopefully we'll have Jut joining us backstage. Jut is where is he right now? He's in Arizona, and he will in a couple days be making his way on quite a long journey to get to Rapa Nui. And then we'll talk about his science, which is really exciting. And there's the possibility of maybe even discovering a new species or two. So we're really excited to connect with Jut. But for now, let's get this video queued up and let's see what the Ocean Conservation Trust has in store for us this week.
Hello again everyone, my name is Georgia and welcome back to the Ocean Conservation Trust here at the National Marine Aquarium in Plymouth. and today we're going to show you our wonderful Eddystone tank. So in Plymouth, about 10 miles off the coast, we have got a reef called the Eddystone Reef, and this is underneath the lighthouse out at sea called the Eddystone Lighthouse. And here we have lots of species that we would find on that reef. So in here, this exhibit shows lots of different habitats that you would find on the Eddystone Reef. So we have got some marine plants in there, like kelp, which we would find out at sea. There are rocky reefs, there is the sandy seabed, there are lots of caves, areas for animals to hide and rest. And then some animals, they live in what we call the pelagic area. So that is kind of just more the middle of the water column. That's where lots of our animals spend most of their time. So as I mentioned, there are lots of different local species in here. So that means that these are animals that we would find around the coast of Plymouth. And to find out some of the names of these species, we can use a species identification guide. So over to the side here, we've got a species ID guide that can help us work out what different animals we have in this tank. So it's got all of the pictures and all of the names. So we can either use an animal's common name, for example, a conger eel, or we might be focusing on their scientific name. This is called their binomial name. So that means that it is made up of two parts, and their name is in Latin. So for example, the conger eel binomial name is my favourite because it's just conger, conger. But more complex names, for example, our thornback ray, it's Raja clavata. Now, these two parts of their name come from their classification. So in science, we can group animals based on their similarities, and we can go through a series of classification called the Linnaean classification system. And there's lots of different stages to this classification, but at the bottom, the last two stages are the genus and then the species. So for example, as humans, our species name is Homo sapiens. So our genus is Homo, and then our species is sapiens. So together, that makes Homo sapiens. And it's exactly the same for all of these animals. So if we go back to our thornback ray example, their genus is Raja, and then their species is clavata, making their binomial name Raja clavata. So we can use our species ID guide to the names of the animals in this Eddystone tank. So if we start a lovely pollock swimming past, that's the big white yellow tinge in the middle of the screen right now. These are a very common commercial species of fish. That means people often eat them. And these are the type of fish that is often in fish fingers. So we've got a few pollock in here. Now on the seabed, hopefully you can see one of our blonde rays on the seabed there. And the blonde ray is expert at camouflage. We can see them swimming along there, and their colours help them to blend in with their habitat. So this would help them to hide from any predators lurking around the reef. And in the middle of the screen, hopefully you can see a very bright blue and orange fish. Now, a lot of people in the UK think that we only have boring brown and grey fish in our seas. But this fish is a cuckoo wrasse, and that's the male. Now, the males are bright blue and orange in colour, but the females, they are slightly different. They are orange in colour with black and white stripes going down their back. And all cuckoo wrasse are born as females. But when they get to a certain stage in their life, some of them will transition into being males. And this is called hermaphroditism. So that is a super cool fact about the cuckoo wrasse. And in the middle of your screens now, we have got a smooth hound, a common smooth hound. So in the UK's waters, we have about 40 species of shark, and in this tank, we have 4 of those. So we've got that common smooth hound, and then we also have some starry smooth hounds. They look very similar, but they have lots of little white spots. Makes them look like they have the stars in the night sky spotted over their backs. So resting on the rocks behind the kelp, there is a bull huss, and this is another type of shark which we have in the UK's waters. Now these sharks, they look very similar to cat sharks, but they're quite a bit bigger, and they've got 2 little fang-like structures on the front of their face, which almost makes it look like they have little fangs, but they're actually called barbels, and that is a really distinguishing factor to help you tell apart a bull huss and a cat shark. Cat sharks, on the other hand, are much smaller in size, and the bull huss and the cat sharks, they are 2 species of shark that can lay egg cases. Now again, we've done a whole other section for the world's most exciting classroom about egg cases and what they are for, so if you're interested in hearing more about that, then you can go back and watch that episode. But we have some egg cases in this tank, which I'm going to show you. So up on the rocks, tangled around the dead man finger coral, there is an egg case that's been laid by one of our cat sharks or our bull huss sharks. And what the cat sharks and bull huss sharks do is they will tangle the ends of the egg case around these corals so that they stay in place and they don't wash away. So it keeps them nice and safe. And then because this is an aquarium, not the middle of the ocean, what happens is when our divers go into the tank, they can take these egg cases out and move them to our nursery where they can take care of them whilst the babies develop, and after 6 to 9 months, the babies will hatch out of these egg cases. So in this tank, we've met lots of animals that camouflage against their habitat. So be that maybe some of the flatfish or the rays that live directly against their habitat, and their colours blend in with where they live. However, some other animals in here that we described as pelagic, they spend a lot of their time swimming in the middle or the top of the water column, making it harder for them to blend in against their habitat. Now instead, they can have a different type of camouflage, and this is called counter shading. So if we focus on some of our pelagic animals, like our grey mullet or our stone bass, you'll notice that they have got a lighter underside and a darker back. And this is very similar to animals like orcas. So an orca is another name for a killer whale. And we've got some models of these hanging from the ceiling. Now orcas have a very big contrast between their dorsal side, their back, being black, and their ventral side, that's their tummy, being white. Now this helps them to blend in, because if you are a prey animal swimming underneath them and you're looking up at their body, their white tummies will blend in with the sky, with the light coming from the sky from above them. But if you are a prey animal swimming above them, looking down on them, then their black backs will blend in with the darkness coming from below them from the deep sea. So this helps them to camouflage and then sneak up on their prey to be able to catch them. So hopefully you've enjoyed meeting some of our local species in our Eddystone tank today. Now, as I mentioned earlier, lots of these animals are commercial species, meaning that we might eat them. So to make sure that we are doing our bit to look after these commercial species, if you eat fish, a really simple thing you can do is making sure that you check where your fish has come from and whether it has been sustainably caught. That means checking where it's been caught and checking what fishing methods were used to catch it. Now, there is something called MSC certified fish, and this means all that checking can be done for you to check whether the fish you are buying and eating is sustainable or not. If you wanted to do any more research, you can go to the Good Fish Guide, and that can help you work out whether the fish you're choosing to buy and eat is sustainable. Thank you very much for watching. We hope you learned some bits about our Eddystone tank, and we'll see you again soon. Bye!
All right. Well, huge shout out to Ocean Conservation Trust. Thank you for that amazing tour of the Eddystone tank. I've got a Kahoot quiz ready to go. We learned a lot of cool things there about this different kind of reef. So more of a stony reef with coral and different species of fish. And then we learned a little bit about commercial fish. So the fish that we like to catch to eat. We learned a little bit about scientific names and why those are important. And then counter shading, which is pretty cool. We've encountered a lot of animals, a lot of species on our journey so far, like penguins and sharks that use counter shading, where they have light underbellies. So if a predator is looking up, they blend in with the sky. But then there's also the darker on top. So a predator looking down, they kind of blend a little bit more into the bottom. I think, Grant, are you trying to get my attention? Should I come visit you for a sec? Nope. Okay. All right. It looks like they're still tuning in on the ship, and it looks like the sun is just starting to come up. Those lucky characters are having a great ocean sunrise right now. Very cool. Yeah, let's take a look at that sunrise. There we go. There we go, out on the Pacific. Enjoying that sunrise. Things look nice and clear. Very cool. Thanks, Grant. All right, very cool. Another sneak peek live on the ocean with the Oosterschelde. So let's get into that Kahoot action. I'm going to share my screen again. Make sure I've got the Kahoot started. That's important too. There we go. And we're going to share my screen, and we are going to get right into the Kahoot action. There we go. Should be loading now. You need to go to kahoot.it. Kahoot.it. And then you're going to want to get this Remember, today's pin is 4609492. I've got 5 questions ready to go. A little bit of true and false, a little bit of multiple choice. And these questions are all related to what we just learned from the Ocean Conservation Trust with the Eddystone Reef. So I'm going to give about a minute to get a few classrooms in. While I'm doing that, I'll talk a little bit about the plan for the rest of the event. So I have a bunch of supplies here for our experiment. It's a pretty easy experiment. There's a few things you need to make, and it's important that today you do have some supervision, whether you're in the classroom or if you're doing it at home, because you are going to need a candle to do today's experiment. We will recap our experiment from 2 weeks ago. If you remember, we did an experiment to trick your brain, where we had 3 bowls of water, hot, cold, and a room temperature bowl. And then we kept our hands both in the hot and the cold. Then we put them both into the room temperature to see what would happen and if we could, in fact, fool our brains. We've got a brand new curiosity, and we're going to spend some time with Jut as well. We'll probably see him in the next 5 minutes. He'll join us from Arizona, and we will do a little hangout together, and we'll learn a little bit more about the science he has coming up. We've got a good group of classes here with us, so let's get our Kahoot going. There are 30 seconds for multiple choice and 20 seconds for true and false. Again, all of these questions you should be prepared for from the last video. So, how far is the Eddystone Reef from shore in Plymouth? Is it 4 miles, 7 miles, 10 miles, or 15 miles? So about how far was that Eddystone Reef from the shore? Was it about 4, 7, 10, or 15 miles? Got a few seconds to get those answers in, and then we'll see who's in the lead. We'll take a little look at our leaderboard. All right, so it is roughly 10 miles from shore. collection of rocks and kelp. Makes a great habitat for fish and other invertebrates like lobsters and crabs and eels and things like that. Yeah. And then there was a lighthouse above it as well, which is kind of cool. Okay, so the daring lemur is in that top spot. Let's go to a true and false question. A commercial species is a species of fish that humans eat. Is that true or is that false? A commercial species is a species of fish that humans eat. True or false? All right, it is absolutely true. Commercial, kind of another way of saying, things that you buy or places you go to purchase things, commercial items, commercial species. So what's really important, if you do like eating fish, like me, I like to scuba dive, but I also enjoy eating fish. You can get those great seafood guides, which tell you which fish are harvested sustainably and which ones you should avoid because they're endangered and not harvested sustainably. So the daring lemur is holding on strong. Let's go to another one. The conga eel, what is its scientific name? Was it conga sapien? Was it conga conga? Was it conga clavata? Or was it conga funebris? So we've got four options there. What was the scientific name for the conga eel? Conga sapien, conga conga, conga clavata, or conga funebris? A couple more seconds to get your answer locked in. All right. Good job, crew. Georgia mentioned it was kind of fun. Conga conga. Not a lot of species names that repeat like that. Our other examples, conga sapien, I took the end from Homo sapien, which is us, the humans. Clavata was from the ray that she talked about. And then funebris is actually from the green moray eel. So conga conga is the correct answer. Daring lemur, you're on fire. 3 in a row. Let's see what happens with our next true and false. Hermaphrodism is when an animal can change its sex. So maybe go from male to female may have been an example shared with us. So what is that, true or is that false? Hermaphrodism, is it when an animal can change its sex? True or false? All right, absolutely true. You do see this in fish, and the picture that I shared there was a great example of the clownfish. Maybe about 2 or 3 months ago, we had a lesson on clownfish from the Ocean Conservation Trust, and then our experiment was making the slime that they have on their skin, which helps protect them from the stinging of the sea anemones, which they use as their home. So we made some slime to kind of replicate the mucus that they have, they can secrete onto their skin. All right, we've got one more question here. True or false? Cat sharks have their young through live birth. True or false? Cat sharks have their young through live birth. True or false? A couple more seconds, get those answers locked in. All right, absolutely false. They have egg cases. So depending on the species of shark, you could have live birth, but there are a lot of species that do have those really cool egg cases. We learned a little bit about them in a past lesson, sometimes called mermaid purses. They come in all kinds of cool shapes, like kind of a rectangle with little hooks on the end. They can be spiraled, so they can kind of be wedged in between rocks. So they do come in lots of cool shapes. But cat sharks definitely have eggs. They lay egg cases. They do not have live birth. Here we go. Let's look at our podium. In third place, we've got the mighty buffalo. Second place, the creative sable. And holding down that top spot, do we have any movement? Ooh, the speedy ibex. Snagging the lead right at the end. Very, very cool. They must have been pretty fast. Okay, let's come back from that screen share And I'm going to share a little link here with you quickly. If you are in the classroom of the Speedy Ibex, I need you to send me an email. You need to send an email to classroom@darwin200.com. There it is. So if you are that classroom, send us an email. We have a gift card to send to your classroom, classroom@darwin200.com. If you are in the classroom of the Speedy Ibex, let's get that email sent to me, and we will keep an eye out for it. All right. I just had a message from Jut. Jut says he's having a little trouble with his Google Chrome. So I'm going to send him a really quick message to see if I can help him out and get him in. Oh, maybe we have him now. I think we do. Here we go. Let's bring him in. Jut.
Hey.
There you go.
Morning.
You obviously did a little troubleshooting and you got in.
Yes, sir, I did. How's everybody doing this morning?
Yeah, good, good. We just had a connection from the ship. They're making their way to Rapa Nui. They should be there about the 3rd is when they're expecting to come in, and they're just preparing right now. They got some crazy winds coming, so the captain's really excited to harness those winds, and he thinks they might get there a little bit early, so that's kind of cool.
Oh, wow. Outstanding.
Yeah. Well, Jut, it's so good to have you here with us. I know it's early for you in Phoenix, but I did want to see if we could get you, because hopefully over the next few weeks, we're going to do some cool live stuff with you as you're doing some really great science in the field. And that's what we're going to talk about today. So first of all, let's get you to introduce yourself, Jut, and tell us a little bit about what you do.
All right. My name is Jut Wynne. I am a research professor at Northern Arizona University in Flagstaff, Arizona. We're at 7,000 feet in the mountains of the Southwest. And in terms of what I do, I am an ecologist, a conservation ecologist, and focusing principally on cave ecosystems. And how I got involved with DARWIN200 is because back when my work on Rapa Nui started in, my goodness, first trip was in 2008. I have been there 5 times since. This will mark my 6th trip to the island. And what we found on our first 2 expeditions was we started turning up these insects, which turned out to be endemic to the island, meaning that they occurred on Rapa Nui and nowhere else in the world. And then we also turned up a couple of insects, which were also known as Polynesian endemics, meaning that they occurred in Polynesia and nowhere else in the world. And this was interesting because a few of these Polynesian endemics were actually we believe, and this is clearly right now a hypothesis, which we are going to be testing on this expedition, is we believe that they were dispersed by the ancient Polynesians. Because when the Polynesians were in their double-hulled voyaging canoes, traversing the South Pacific, traveling eastward from points further west, islands further west, all the way to Rapa Nui, they would carry with them plants in gourds, and they were called canoe plants. And these folks were very smart. What they were doing was they had realized that, well, much like when we do a car camping trip, we want to take all of the creature comforts with us. Well, for the ancient Polynesians, those creature comforts were food. And it was food that they could use for medicinal purposes, for making nets, for making rope, and for food, of course. And they would take these gourds and they would have this, cultivars, small plants within them, saplings, and then they would introduce them as they would move across Polynesia. And through this process, these plants were introduced. And at the time, it's not like there was a Department of Agriculture that would inspect what they were introducing. And when they were doing that, they would also be introducing the insects that were within the soil. And we believe that there are at least 3 species thus far that have been introduced in this manner. So we're going to be looking for those animals, as well as potentially other Polynesian endemics that might be canoe bugs. And also we're wanting to potentially debunk this hypothesis as well. So we're looking for other alternative explanations We hope to be sampling the open ocean, to be looking for floating or rafting terrestrial insects. And we're also going to be keeping an eye out for rafting debris, like big branches or coconut palm trunks, that sort of thing, so we can see if there are any arthropods floating on on that debris.
All right. Very cool, Jut. I'm very excited about this science because it's good science. We've got a great hypothesis to test where there's some evidence for a theory one way, but the thing about science is sometimes the hypothesis is wrong, and that's a valid part of science too. That's why we go out, we ask these questions, and then we usually come back with a whole bunch more questions. So I'm sure you're getting pretty excited to get out there.
Oh, indeed, indeed. This is an opportunity of a lifetime for me to actually test this hypothesis. I've as science typically works, we take maybe a step forward and a couple of steps back, and it just moves. Science does move very slowly for the most part. Sometimes we do have these big leaps and bounds in terms of making progress and discoveries, but what we're seeing here is an opportunity to actually test this idea. And like I said, at the same time, to look for alternate explanations as well, because we don't want to become, I'm not, it's an interesting idea. It's certainly compelling and exciting because we're tying, ancient culture to how animals may have dispersed. But at the same time, we got to be wary and say, well, there are other possibilities, and we got to equally ferret those out as well.
Absolutely. So classrooms tuning in, if you do have any questions for Jut as we're having our conversation, please put them into the chat sidebar. I'll keep an eye out for those. But I want to ask you, because we talked about Rapa Nui, but you have explored caves in places all over the world. Can you tell us about some of the other places you've been?
Oh, shoot. Yes. So I have worked most recently, been working in Belize. I've been down there past few years. Golly, let's see. We're working on 4 or 5 research trips down there now. And one of the things that we're doing is we're looking at novel animal behaviors in caves, specifically by using trail cameras to monitor animal behavior. So we're putting these trail cameras in these caves and letting them run for an extended period of time. And in this case, we have in 2 areas, we have cameras have been running for at least 1 year. And in another area, we are looking at almost 2 years of collecting data. And we have some really interesting results. We're getting a lot of jaguars. We're getting, we have the first evidence of jaguars actually using caves as a feeding site. We also have a bare-throated tiger heron that was actually hunting or fishing, as the case may be, underground. So we're really making some contributions in that regard. That's been really exciting work. We're also working on, we're studying bats down there. I spent last summer getting bit about every night. It's interesting because I've worked in the Southwest for so long with bats, and comparatively speaking, bats up here are very docile compared to bats down in Belize. They're all teeth, and they're just like, when you have them in your hand, they're like, and they're just constantly trying to bite you. So as a result, I got bit quite a few times. So in Belize, I've done a lot of work, as I mentioned, China, Spain, where else? Hawaii, throughout the American Southwest. I think that's it. Maybe not. But yeah, for right now, it's pretty early in the morning, and I haven't had a lot of coffee in me, so I'm kind of, waking up.
No worries. That's a pretty good list, Jut. But your explorations of caves aren't just on our planet. Can you tell us about a little bit of the work you've done with NASA?
Oh, yeah. So in way a long time ago, we led a project, 2008 to 2011. We did a study to characterize thermal behavior of caves, and we did that by collecting temperature temperature data using these HoboPro data loggers, and we deployed them in caves in the Atacama Desert in northern Chile. Hey, also worked in Chile, and the Mojave Desert of Southern California, and the project culminated by us going in and or flying a NASA aircraft rather over the Mojave Desert. And we collected thermal imaging, and we use this thermal imaging because thermal imagery, what you can see is if you were looking at me right now in a thermal image, I would glow like red and orange colors, and everything behind me would be probably these bluish colors because it would be cooler. The background would be cooler and I would be warmer. So that's what the same principle holds for caves. The entrance was either cooler or warmer than the surrounding terrain. And we actually did a paper on that a while back. And since then, and the reason why that's so important is because we're using these as using those sites as what we call Earth analogs, because we believe them to be analogous or similar to places on other planets that might support caves. And right now we have found cave-like features in 11 different planetary bodies across our solar system. And there are what we call speleogenetic processes, in other words, cave forming processes that have been documented from Mercury all the way to Pluto. And that's super exciting. We've done several papers on that. I've collaborated with NASA folks and European Space Agency folks from all over the world collaborated with us on these studies to inventory caves across the solar system.
All right. Very cool, Jut. We've got a question here for you. Working in the caves, have you ever gotten lost in a cave before?
Oh, ever gotten lost? I would not say lost. I would say I've been turned around on a couple of occasions. We ultimately do find our way out. We do have cave maps, and once you've done this for a while, you do develop, I guess, a fair degree of acuity in terms of knowing your way around, recognizing landmarks as you're moving through a cave, so that you can orient yourself so that if you do get turned around, you can figure your way out. What does happen, being a scientist and basically being a 10-year-old kid in an old guy's body, is that you see things and you get excited when you see things. And you're like, oh, look at that. Oh, look at that. And when that starts happening, you can get turned around. And that's typically when those occasions have occurred. When I get turned around, I get excited. And then all of a sudden I realize, oh, where am I? Oh, okay. And then I have to figure out where I am.
All right. What's the most interesting species you found in a cave?
Interesting species. Ooh, that's a good question. Wow. Well, I guess, are we talking big or are we talking small? If we're talking big, for me, jaguars. We've documented jaguars quite a few times in caves. We found evidence of jaguars on numerous occasions. That the example that I provided earlier about documenting jaguars using caves as a feeding site. We were working in this one cave in Belize, and we're about 300 feet underground, about 100-ish meters, and we're underground, and all of a sudden we see jaguar poop. And we're like, oh, wow, okay, jaguar poop, that's interesting. Well, at the time, we didn't think it was jaguar poop. We just knew it was really large carnivore. And with jaguars being the largest one down there, we were like, yeah, it's probably jaguar. It could be mountain lion, or what they call puma down there. But it was like, we're probably pretty certain it's jaguar. And then we found a bone scatter. And then we realized at that point that it was a peccary, because we found the skull, big canine holes through the top of the skull. And we were like, wow, this is super neat. And then later, a couple of weeks later, we pulled the trail camera footage. And then we're like, ooh, definitely jaguar. So big animals, jaguars. In the fall, I'm going to be going to Bhutan with a similar objective. But at this point, we're going to be looking for tigers. So big cats are certainly really cool. You don't want to encounter them in a confined space. That would not be good for the researchers potentially. But collecting the footage and finding the evidence, super exciting. Very small animals. I think perhaps one of the neatest ones that I keep leaning on is we found in China, in southern China, what are called dragon millipedes. And those are super neat because they have like these spines along their back, which is how they got their name, because they kind of look like dragon spines.
All right. Very cool, Jut. All right. So how many days do you have, Jut? When do you take off? I think on the 2nd. Is that right?
I'll leave on the 4th.
Okay.
And it's overnight, very fun flight down to Chile. And then from there, I got a very tight layover, and then boom, head out to Rapa Nui from there.
Yeah, we're keeping our fingers crossed. Things go smooth on the tight layover. I'm sure we'll get you there one way or another. But we can't wait, because the next time we connect with you, it should be from Rapa Nui, and hopefully we can see you in action in the field.
Yes, that would be wonderful.
Awesome. Well, Jut, thank you so much. I know you've got lots to do to prepare, so thank you for getting up early and hanging out with us. Get a few more cups of coffee into you, and I'll talk to you soon.
Joe, thank you. And thank you, everyone. Pleasure to be here. Y'all take care.
Jut. See ya. Okay, there we go. So that's Jut Wynne. We will see Jut over the next couple of weeks. With any luck, he'll be doing some great collection of specimens in Rapa Nui. Pitcairn, we're going to travel to Pitcairn on the way to Mangareva. Now, it's 50-50 whether we're going to get to stop at Pitcairn, because it doesn't have a really good harbor. It's a very small island. If weather conditions are good, we stop for the day, and Jut can do work for the day. If weather conditions aren't good, we have to keep on going and keep making our way towards Mangareva. And then on Mangareva, Jut will be doing the work again in the caves, looking at invertebrates, collecting samples of different species he finds. And two things are going to happen. We're going to look at that canoe bug theory. Were Polynesians moving those invertebrates, those insect species, from island to island? So that's one part of it. That's going to be pretty cool. And then possibly, we can't say for certain, but there is a chance we could find a new species or two, an endemic species, so only found on one of those islands. That would be really, really cool to be able to do that. So we'll see what happens. Stay tuned over the next few weeks. So we have to talk about our experiment from 2 weeks ago, and then we're going to do our brand new experiment before we wrap up today. So I'm going to share my screen one more time here just to remind you of our experiment from 2 weeks ago, where we tried to fool our brain. We have an experiment for this week, and I picked a really, really easy experiment. Turn the volume down there because we don't need the volume, but we do need to be able to fast forward a little. So that's what I'm going to do. So with this experiment, it was probably the simplest one we've ever done. We had 3 bowls. In one bowl, we had really cold water. In one bowl, we had really hot water. Not so hot you couldn't put your hand in it, of course. And then in the middle bowl, we had room temperature water. Now, what we did is we put a hand in the cold water for 30 seconds, and we put our other hand in the hot water for 30 seconds. Just kept our hands in there. From there, we took both hands out and put them into the room temperature water. And then what did we feel? Well, what we felt was our hand that was in the cold water felt very hot when we put it into the room temperature water. And then our hand that was in the cold water, our hand that was in the warm water felt very cold when we put it in the room temperature water, which is strange. It's a complete reversal of what our hands were just feeling, especially when we put it into water that should have just been kind of room temperature. So not warm, not cold. So what was going on there? How did we trick our brains? Well, we've got thermoreceptors in our hands, well, all over our body. But in particular, we were looking at our hands this time because we had our hands in the cold, our hands in the warm. So we really activated our warm receptors in the warm water and our cold receptors in that cold water. So we really confused our senses when we took our hands out after 30 seconds and we put them right into that room temperature water. So our thermoreceptors aren't thermometers. They don't say, this is 30 degrees Celsius or this is minus 10 degrees Celsius. They kind of measure relative. So, this is cold, this is warm. So when we primed our one hand to be thinking about really warm with the warm receptors and our other hand to be thinking about really cold with the cold receptors, then we put them both into that water. Our warm hand felt relatively cold because it moved into the room temperature water, and our cold hand felt relatively warm because it moved to the room temperature water. And so that's an evolutionary response. It's really valuable for us to be able to feel temperature changes and react to them. For instance, if you're in the kitchen and you're cooking and you accidentally put your hand on a hot pot and you pull your hand away really quickly. So your body doesn't really ask questions about what temperature is this? Can I keep my hand here? You react. Your warm receptors react. So that's an evolutionary thing. We're primed to react to these temperature changes. And so that's what we did with our body. Even though the water in the middle wasn't hot, it wasn't cold, it was just regular old room temperature water, to our hands that were in really cold water and really hot water, it felt extreme. It felt like the really hot hand was suddenly feeling very cold and the really cold hand was suddenly feeling very hot. So that was our experiment where we tricked our bodies with those different water temperatures. And so again, it's relative. It isn't hot water, it isn't cold water. We don't measure in that way, but our bodies are primed to respond to that change in temperature. So it felt much more extreme than it actually was. All right, let's do an experiment together. Let's see how it goes. I'll take that old video out and I'm going to switch to a second camera. There we go. Let's get this camera full screen. All right, that should look nice and full screen. And let's clear a little bit of space and let's do this experiment together. And with any luck, it'll work really smoothly. Live science, you never know. Sometimes it works really well, sometimes not. So for this experiment, a few things. You need some paper. So preferably like computer paper. So construction paper is a little heavy. You want just computer paper for this. You want a pen. You want some scissors. Skewer, like a bamboo stick, like this one here. It's kind of sharp at one end. Some plasticine, a little ball of Play-Doh or plasticine. What else? A candle. So I've got just a little tea light here, nice and easy. And then something like a lighter or a barbecue lighter, matches, whatever, something so you have a little bit, you can make a little flame to light your candle. So let's get going. I'm going to take my pen and I'm going to draw a spiral on this sheet of paper. Let's mess around with that a little bit. So I'll start in the middle and I'm going to build my way out. I'm going to make it pretty thick. And it's up to you how big you go. You'll see why you don't want to go too big. If you go too big, you're going to run into a problem. And we'll talk about that problem if we get there. And then just kind of end your spiral, go right in to that last spot. So I've got my spiral. I'm going to grab my scissors and I'm going to quickly cut out this spiral. So first I'll go around the whole thing. So there we go. I've gone around. I'll get my paper out of the way. And then I'm going to carefully cut out my spiral. So I'm just going to follow this path that I made all the way till I get to the middle. I may have made mine a bit too big. I might need to, excuse me, cut it a little bit, but that's okay. We'll talk about why it can't be too big. I think you'll be able to figure it out pretty quickly why it can't be too big. So take my plasticine. That looks like a good spot. Make sure we can see it on camera. Put it right there. And then I'm going to take my bamboo skewer and I'm going to put it right there like that. Maybe change my camera angle just a little bit so I can see the top. There we go. And then I'm going to take my spiral. You'll see it kind of hangs down like this. I'm not going to poke a hole in the middle, but I am going to kind of just bend it over the top a little bit, just so it sits a little bit there. So it's sitting on top. I didn't poke a hole in it. It's just sitting on top. I'm now going to take my candle and I'm going to place it next to there. And then I'm going to take my lighter and light the candle. So I think you can probably guess why you don't want your spiral to be too big. If your spiral's too big, it'll catch on fire. So make sure your spiral is not so big. There we go. All right, so I've lit the candle and now my spiral paper is turning. Very cool. You can also, if you want, I've got a second one here. You can also do the same thing, but put it on string instead, like this, and you can hold it over top of the flame, your candle. So that'll work too. I like the bamboo skewer. It's pretty cool. You can see I made mine slightly too long. It's almost reaching the candle, but we can blow the candle out now because I think we've seen pretty well how this works. So there we go. That is our experiment for this week. All the tools are here. If you need to go back in time and watch this, we will have it in a playlist on the website for you shortly. But that is our experiment with our spiral paper, with our candle, and then we had our spiral start to spin. So I would like to see, let me switch camera views here. There we go. So I would like to see, we would like to see you try this experiment. Maybe you're going to try it at home. Maybe you're going to try it in the classroom. I will share the link here. If you try it, if you take some photos, send them to classroom@darwin200.com. Oops, that's the link to the website. That's important too. You can check out the map and other things at darwin200.com. But we want you to send some pictures trying this experiment to classroom@darwin200.com. And then next week, we'll tell you what happened and why it happened. But we'd like to hear what you think. How come this spiral began to spin when we lit the candle beneath it? We want to hear what you're thinking about this experiment, why you think we had the results that we had. So send in some pictures. If you have a video clip, that's cool too. But please, please, please make sure you do this experiment with a parent, a guardian, or if you're doing the classroom with supervision from your teacher. So there we go. We need to wrap up now. There's 2 weeks to get that in. We need to wrap up with our curiosity of the week. So here's a video checking out last week's curiosity. Let's see what last week's curiosity was.
Back everyone. Last week I asked you to try and identify this intricate mineral formation. What do you think this object is and how did it form? Well, it's known as a desert rose. They develop in dry, arid, sandy conditions. They're particularly well known from the Sahara. They actually form of gypsum and other minerals that grow out in these amazing plate-like structures and formations. Some of them really can grow just like roses in a circular pattern, look almost like flowers developing in the middle of the desert. So if you guessed desert rose, you are absolutely right.
All right, so there we go. Thank you to Stu for filming that from the Darwin studio. This week, our new curiosity, I'm going to do here from my office because Stu's been bouncing around a little bit as we get new parts of the expedition together. So I've done the last few experiments from the office, and we'll do this week's curiosity from the office as well. With any luck, by next week, we'll start doing some new experiments that will be filmed from the Darwin studio. when Stu is back in the UK for a little bit. But for this week, we have a object here with me. Now, I think most of you will take a look at this right away and say it's a rock, and you would be absolutely right. But that's not what I'm asking you. I'm not asking you to tell me it's a rock, because I just told you. What I want you to try and do is two things. Let me get this a little closer to the camera. There we go. I want you to tell me what kind of rock this is. So that's step one. Can you tell me what kind of rock this is? The second thing I want you to tell me is, it's a little hard to see in the camera. Sometimes it gets a good focus. There's little green crystals in this rock. I want you to try and identify what these little green crystals are in this rock. So don't tell me it's a rock. We know it's a rock, but I want you to tell me what kind of rock you think it is. And then I want you to try and identify the mineral in this rock. What are these little green crystals in the rock? And I'll give you a little clue. This rock is from an island in the Azores, in the middle of the Pacific Ocean, and we will be visiting the Azores in a long time from now, all the way next year around June, July 2025. So what kind of rock is this? And can you identify the mineral that makes these little green crystals in the rock? So you have 1 week for this one, for the curiosity, classroom at DARWIN200.com, 1 week for classroom at DARWIN200.com. And there we go. That is the end of the world's most exciting classroom this week. It was another busy one. We bounced around the world. We met new people. We met some of the Darwin 200 team on the ship. And now we have our challenges. We have our brand new experiment, and we have our curiosity of the week. We will see you next week when we will be live from Rapa Nui, or Easter Island, as maybe more familiar to some. And hopefully we'll be out in the field and show you some pretty cool things on this amazing island, part of Chile, way out in the middle of the Pacific. So what we need to do to wrap up, as always, we need to thank our sponsors, our supporters. So that's what we're going to do with a little video clip here. Without them, we wouldn't be able to do the world's most exciting classroom, to sail around the world, to train young conservationists, to do cool research. So our partners, our sponsors, we are so grateful to them. And here is a little thank you to wrap up today's world's most exciting classroom.